Welcome. In this video, we will cover the respiratory system in short clips. We will first look at the anatomy of the respiratory system, and then begin to cover what exactly happens during inspiration and expiration. So let's go to the slides. All right. The respiration system has three functions. We are normally aware of one function, which is gas exchange. So it essentially will acquire oxygen from the environment through the lungs, and it gets the oxygen in the bloodstream and then takes it to the doorstep of every cell. And then the cells will get the oxygen. And in exchange, the byproduct of metabolism, cellular metabolism, one of the key byproducts is carbon dioxide. And so the cells will release carbon dioxide in the bloodstream, and then it will be taken to the lungs, transported to the lungs, and then from the lungs it goes to the atmosphere. So this is really the best known function, which is the primary function, gas exchange or respiration. But the respiratory system also has two other functions. One of them is pH regulation. It's not the primary system that regulates pH. The primary system that regulates pH is the renal system. But the respiratory system does play a role in pH regulation. The third function is temperature regulation. In human, it's the skin that's the primary system that regulates temperature. It's the skin that's the primary system that regulates temperature. But in dogs, it's exclusively the respiratory system. And so that's why when you're taking your dog on a walk, it will stick its tongue out and will be panting, because dogs do not have sweat glands like you and I. So in this lecture, we will focus largely on the gas exchange function. We'll touch a little bit on the pH balance or acid base balance, but we will not talk about temperature regulation. And so to begin the gas exchange, we go to this slide, and gas exchange is divided into two parts, or respiration is divided into two parts. You have the external respiration and internal respiration. What's the difference? Internal respiration is essentially aerobic. It has multiple names. So internal respiration, cellular respiration, aerobic respiration. It's basically the same thing. And it basically just means the use of oxygen to produce ATP. As you are aware, there's anaerobic. Anaerobic is without oxygen, and aerobic is oxygen dependent of ATP production. And that happens in the cell. This is happening inside the cell. The rest of it is external. And so please don't confuse external respiration to ventilation or breathing. Ventilation or breathing is a part of external respiration. But external respiration begins from the acquisition of air into the lungs, transportation of air in the blood vessels right up to the capillary. So that's external respiration. And so let's begin with external respiration. And let's track the gases as they move, which is essentially oxygen and carbon dioxide. And then we'll go into more details later. So begin with oxygen that's acquired from the atmosphere. So oxygen goes in the lungs up to the alveolar of the lungs. That's the site for gas exchange. And again, like we've said before, almost everything in the body moves down the gradient, down the slope, whether that's air or blood or molecules or ions, they move down the slope. And so there's a gradient then, which we shall talk about later, that's pulling oxygen from the atmosphere into the lungs at the alveolar level. And then again, there's another gradient between the alveolar and the pulmonary capillary, and then oxygen then will move in the blood from the lungs. It goes to the left atrium and then left ventricle into the aorta and the rest of the arteries right up to the capillary next to the cell. And oxygen again is using a gradient to move from the capillary into the cell. And then after cellular respiration, carbon dioxide is made, and carbon dioxide will move down the gradient back into the capillary, and then will be carried will be transported to the big veins, right atrium, right ventricle, pulmonary trunk, pulmonary capillary, into the alveolar, again using gradient, and then out into the atmosphere using gradient. So that's the route. That's the route that the gases are using. Okay, so let's look at the gross anatomy of the respiratory system. And so when we talk about the respiratory system, we include more than just the lungs. So we start from the nose, okay? The nostril, the nasal cavity, the mouth, and then the pharynx. This is the pharynx, and then the larynx, where the sound box is. And that's one thing I forgot to mention, maybe the fourth function of the respiratory system is sound production. Sound production. And the larynx produces the sound. And then from the larynx, we go to the trachea, and then the trachea is divided into the right and left bronchus, and then that increasingly then will branch into further smaller windpipes until we reach the alveolar that we talked about, okay? And then around the lungs, you have the ribs, and between the ribs, you have muscles that we'll talk about soon. And then in the center, between the two lungs, you have the heart. And then you have the demarcation of the thoracic cavity. So that's the thoracic cavity. And then below the thoracic cavity, you have the abdominal cavity. That's where you have the stomach and the intestines. Now, the major landmarks that you should pay attention to, the diaphragm. We've already talked about the diaphragm before. Pay attention to the ribs and the nasal cavity. Let's keep going. Okay, so in the previous slide, we talked about more about the gross anatomy, but here now we go into the last branching, the terminal branching of the windpipe. So we've already talked about the alveola, and they look like grapes. They have a grape-like structure, which allows for increased surface area. So if you were to flatten, so you see these cavings, these increase the surface area. If you were to flatten the whole surface of the alveola, it would be the size of a tennis court. So this is massive increased surface area. And just before the airways get to the alveola, you have the bronchioles. We've talked about this quite a bit when we're talking about the autonomic nervous system. You have the bronchioles, and the bronchioles have smooth muscles. So you can see the smooth muscles in the bronchioles that allow the changing the radius of the bronchiole, which then affect the airflow. Okay, And then the other thing I want you to pay attention to is the relationship between the pulmonary capillary, which is here, the close relationship between the pulmonary capillary and the alveolar. In fact, the distance, as we shall see later, the distance between pulmonary capillary network and the alveolar is just 0.5 micrometer. So very, very thin. Remember, you did talk about the factors that affect diffusion rate. So surface area is one. So here you see classical conditions of an infrastructure that promotes high diffusion rate. So you have an increased surface area, like I said before, because of the structure of the alveolar. And then the other factor that affects diffusion rate is the membrane thickness. So the distance between the capillary, the pulmonary capillary, and the alveolar is very, very thin, very, very small to promote diffusion. And then the other factor, which we shall see later, is the gradient, the steepness of the gradient, and also temperature. So the air that's coming in is warm and wet because of the nozzle. The nozzle cavity makes the air wet. It's not dry, but also warms the air. Okay, so with that, let's go to the next slide. Okay, so we're now going to focus on the alveolar. Okay, so we have a cross section. Sorry about that. We have a cross section of the alveolar, and you can see these inward extensions that increase the surface area. And there's a disease that can compromise, reduce the surface area, which is caused by smoking, emphysema. Emphysema basically collapses some of these inward extensions and therefore reducing the surface area and subsequently reducing the diffusion rate, making it hard for the patient then to breathe. Okay, so let's go down here on this diagram. So we're going to expand this part here. You can clearly see the last part of the bronchiole here. Let's expand here so that we see the close relationship between the capillary. Sorry about that again. So you can see this is the capillary. You can see the red blood cells there, and you can see the endothelium. We've talked about this, the 1 cell thick lining, the inner lining of every vessel, including the capillary. In the case of the capillary, that's the only lining which makes these vessels ideal for exchange, for diffusion. Okay, and then you can see the close interaction between the capillary and the alveolar. So this is about 0.05, like we said, micrometer, very, very thin membrane to promote diffusion. Okay, let's look at the alveolar. In the alveolar, we see different types of cells. Here we see type 2 cell. And on this other side, we see type 1 cell, and we shall see a third cell type. So there are 3 cell types in the alveolar. In the next slide, we're going to see these cell types. Okay. All right, this is again the distance between pulmonary capillary and alveolar, 0.5. In the previous slide, I said 0.05, I misspoke. It's actually 0.5 micrometer. And there are diseases that can increase this distance, and because of inflammation and infiltration of fluid. And so if you change this distance, then you compromise on the diffusion rate, and that can become very deadly. So a disease like pneumonia can change this thickness and therefore become extremely dangerous to the patient, because then the diffusion of oxygen is compromised. Okay, so let's focus now on the alveolar. Like I said, we've already seen type 2 alveolar cell and type 1. And then the third is the alveolar macrophage. So let's look at each one of them and see what they do. All right. Type 1 alveolar cells basically just form the cell wall of the alveolar. Okay, so that's easy. Just forms the one cell wall, 1 cell thick, again to promote diffusion, to make a membrane very thin. What about type 2 alveolar cells? Type 2 alveolar cells produce a fluid that's called pulmonary surfactant. Pulmonary surfactant. We shall come to that. This is a very important fluid that makes it easy for us to breathe. How does that help us to breathe? And the illustration I use is, imagine when you were 2-year-old and you just loved balloons, and each time you went to the shop with your mom, you'd go to the balloon section and grab a balloon and you try to blow it, and your mom would say, honey, let me help you. And you'd say, no, I'm old enough to do it. And you'd try to blow it and you'd fail. So blowing an empty balloon is like breathing without surfactant. It's very hard. And so thanks to the surfactant, it makes our breathing effortless. And there's a disease, the disease that affects premature babies, because the lung and the brain are one of the last systems to develop. So if a premature baby is born before type 2 surfactant develops, then there will be no pulmonary surfactant, and the preemie babies will have difficulties in breathing. And so the OBG therefore has to predict and foresee that this mother is in danger of having a premature baby, and the solution is easy. They just have to give the mother the stress hormone cortisol, because it's cortisol that helps these cells to mature and produce surfactant. And the disease is respiratory distress syndrome. So again, just think of blowing the balloon, an empty balloon. That's how the premature babies feel like. So what exactly then is the surfactant doing? The surfactant, to make it easy, goes, in the absence of the surfactant, you have a water film consisting of water molecules. And because of the electrical charges of a single water molecule, they tend to create an attractive tension between each other. And again, to make this easy to understand, imagine you create a circle and you hold your hands in a circle, and you are each water molecules. And then if everybody is pulling the people that are standing on the on each side of their hands, if you pull everybody towards you, the circle will become smaller. So the force will be towards the center. Okay? And so if you just have water molecules without the surfactant, the alveolar will create a centripetal force that will go towards the center, therefore making it hard to expand when you're trying to breathe. So the surfactant then will go between the water molecules and tone them down, and therefore reducing the force to the center, and therefore making it easy then to breathe. So that's what type 2 alveolar cells are doing. What about macrophage? Macrophage is like a sentinel. Remember that the alveolar interacts directly with the outside, with the environment. Okay, so think of the alveolar as a border center. And so macrophage is there to control, to prevent entry, illegal entry into the body. So it's controlling entry of bacteria or microbes. All right, so with that, let's go to the next slide. So this slide then talks about the actual breathing. So here now we start breathing, which is part of the external respiration. So ventilation is divided into inspiration and expiration. Inspiration is breathing in, expiration is breathing out. Now, again, remember what we said. Things in the body move based on gradient. And so when you're breathing in, during inspiration, it means that there's a gradient. There's a gradient for air to come in. Why? Because the partial pressure, which is P, we're introducing a new term, the partial pressure of the alveolar then becomes less than the partial pressure of the atmosphere. So the pressure in the atmosphere will be greater than the pressure in the alveolar, and therefore air will go in. There'll be a positive flow. I'm sure you're asking how is the gradient created? We'll look at that. But for now, just know that there's a gradient that pulls air inside. And then during breathing out, expiration, the gradient is switched. It goes to the opposite side. And we'll see how that's happening. This is a special case where a gradient is flipping, is changing. So during inspiration, it's one way, it's inward. During expiration, is outward. And we shall see exactly how that's happening. So this brings us to an equation to explain what's going on here. So airflow, which we talked about in the blood vessels, it's the same principle. So airflow is equal to delta P divided by resistance. Delta P is basically showing the steepness of the gradient, okay? The pressure difference. Delta P is the pressure difference, so which shows the steepness of the gradient, and then R is for resistance. So in other words, if we expand this, is the pressure difference between the partial pressure of the atmosphere minus the partial pressure of the alveolar. That gives you the steepness. Okay, so that gives you a steepness, and you divide that by resistance. In other words, then, if you were to draw a slope, so explaining this formula in simple terms, if we were to draw a slope and we give an angle of that slope, then resistance then would be, we throw some stones on the slope, and the stones on the slope can affect flow. So you can have the same steepness of a gradient, but one gradient is smoother than the other, and so that's the R. We'll go into more details on that. So let's now zoom in. Let's go into the details of inspiration and expiration. Okay, we start with inspiration. Now, before we go into inspiration and expiration, let's introduce one law that we talked about in the cardiovascular system. We didn't give it a name. Now we're going to give it a name. You may have encountered this in physics and chemistry. And so that's the Bohr's law. The Bohr's law talks about the relationship between volume and pressure. Remember, here we were talking about the systole, during systole and diastole, the volume of the ventricle change. During filling, the volume expands and the pressure drops in the ventricle relative to the atrium. And during systole, as the ventricles are contracting, the volume decreases and pressure increases. So there's an inverse relationship between volume and pressure, okay? So as you increase the volume, the pressure drops, and as you increase the pressure, the volume will drop. So using the same principle, this is how inspiration and expiration are regulated. So in this case, we're really talking about thoracic cavity, the volume of the chest or of the thorax. So you may ask, but what's changing the volume? Well, there are two major muscles that are changing the volume of the chest in 3D, in 3 dimensions. So the diaphragm increases the volume in a vertical way. When the diaphragm contracts, it expands the volume in a vertical dimension. And the inspiratory muscles, when they contract, they expand the volume sideways and forward. So collectively, therefore, they expand the thoracic cavity in 3D. And based on Bohr's law, when the thoracic cavity expands, the pressure in the thoracic cavity will decrease. And the decrease in thoracic cavity pressure then creates a gradient. So the pressure, atmospheric pressure then, the partial pressure of the atmospheric will be greater than the alveolar, or putting it in another way, the pressure in the alveolar when the thoracic cavity expands becomes less than the atmospheric pressure. And that then causes a positive flow of air into the lungs. Again, based on our formula here. And in expiration, the opposite is happening. Again, based on Bohr's law. In this case, then the diaphragm and the inspiratory muscles will simply relax. They go back into their original position. So for the diaphragm, it decreases vertically. And then for the inspiratory muscles, as they relax, the thoracic cavity then decreases sideways and forward and backwards. And therefore the volume of the thoracic cavity decreases, and based on Bohr's law, the pressure in the thoracic cavity then increases, and therefore the partial pressure of the alveolar is greater than the pressure of the atmosphere. And so air then will move from the alveolar out into the atmosphere. Okay, so let's now look at the actual thoracic cavity and see where these muscles are located. So this is before inspiration, and this is during inspiration. And so you have the diaphragm here, okay, in a relaxed state. And then you have the inspiratory muscles, which we're now going to give a name. The specific name is external intercostal muscles. Intercostal intercostal because they are between the ribs. External because there is an internal. So these ones are on the outside, and on the inside you have the internal intercostal that are involved in expiration. So just remember, external intercostal is involved in inspiration, and then you also have the diaphragm. And so you have the sternum, you have the ribs. This is before inspiration. The muscles are relaxed. And when you breathe, so you see the external intercostal contracting, pushing the thoracic cavity, expanding in a forward, backward way, and also sidewards. And then you see the diaphragm expanding from this original position, going down in a vertical direction. So there's a vertical expansion, sideward, forward expansion, which increases the thoracic volume and therefore decreases the pressure, and that then causes air to flow in. What about expiration? Expiration is passive. There are two types of expiration. There's a passive one where the muscles would just go back to their original position, the external intercostal and diaphragm will just go back to their original position based on the gravity. Whereas there's also active expiration. In this case, you then have two muscles coming in. In the case of active expiration, you have the abdominal muscles coming in, contracting, and you also then now have the internal intercostal muscles. What are they doing? The internal intercostal will just do the opposite of what the external was doing. Remember, the external was expanding. The internal will reduce the thoracic cavity in a forward, backward, as well as the sideway. And then the abdominal muscles will essentially contract to push the diaphragm further up, and therefore reducing the thoracic cavity in a vertical way. Okay, so normally this is done to really increase the steepness of the gradient to have the air going out. And this happens when you're playing sports, you're playing football, you're running, so you have active respiration. All right. In this slide, we talk about lung compliance. Lung compliance, which simply means the ability of the lungs to expand without any hindrance. To expand without any hindrance and without any undue effort. And so there are two factors that influence lung expansion. We have already talked about one factor, the surface tension. So reduced surface tension, and we say the surfactant has a role. And remember the illustration we used, blowing the balloon as a 2-year-old. The surfactant helps then the lungs the alveoli to expand without a lot of effort. The other factor that helps us to breathe for the lungs to expand is there's a protein in the lungs which is like a rubber band, and that protein is called elastin. So elastin makes the lungs elastic. It becomes very flexible, very stretchable, easily stretchable. So these are the two factors that helps the lungs to easily expand. And again, we've already talked about a disease in preterm that could affect surfactant, the respiratory distress syndrome. Here again, you can have another disease that can affect the elasticity of the lung. So things like asbestos. Asbestos is the material that was used to manufacture roofs in the past. In some third world countries, they still have that. So asbestos, if it goes to the lungs, it will cause fibrosis, which then reduces the elasticity of the lung, making it hard to breathe. Okay, and then resistance. We talked about resistance, and it was in our formula. So what are the factors then that affect airflow or resistance? Well, the parasympathetic nervous system, and we've talked about this. The parasympathetic nervous system is a rest and digest. And so when you are resting, the demand on oxygen is low, and so the bronchioles will constrict, so you have bronchial constriction because the demand for oxygen is not much. And the sympathetic will cause bronchial dilation. But we also have talked about histamine. So when you're stung by a bee, the bronchus will go into bronchial constriction. And remember, we said you can give EpiPen to counter that effect. We also have talked about asthma. Okay, with asthma will cause bronchial constriction, and so you use an inhaler that has albuterol, and that will cause bronchial dilation. What we have not talked about so far is the effect of allergy. So allergies, the spring is coming, we're getting into spring. Allergies can also cause bronchial constriction. Okay, we've already talked about epinephrine. It will cause bronchial dilation, and this epinephrine acts on the beta-2 adrenergic receptors, beta-2 in the smooth muscles of the bronchioles, and that will cause bronchial dilation. So these are the different factors that influence resistance and impacts airflow. Okay, so here, this slide is showing the different lung volumes, So I'll go through slowly. So the total lung volume is about 5 to 6 liters, 5 to 6 liters. That's everything in the lungs, which is total lung capacity. And then the lung that's available for breathing is called inspiratory capacity. So this is the air that's available is inspiratory capacity, and the one that's not available for breathing in is functional residual capacity. And the one that's completely not available for expiration or inspiration is residual volume. Residual volume is not available for either inspiration or expiration. The air that's available for inspiration and expiration is vital capacity. So this is available for inspiration and expiration. Okay. Now, the air that remains after normal breathing is inspiratory reserve volume. So when you're relaxed, at rest, and you're breathing, whatever remains in the lung from the inspiratory capacity is inspiratory reserve volume. And the air that you breathe in and out, so this rise is the inspiration and the drop is expiration. So this is the equivalent of a stroke volume, if you may. So that's called tidal volume. Okay, breathing in, breathing out, breathing in, breathing out, and tidal volume on average at rest is 500 mils. Just like stroke volume, it's changing. It's changing based on the metabolic state of your body. So we will stop here.